Good day, my name is Ivan Tюmin, next to me Nikita Kazarin. We are the founders of biotech company Y-combinator. Today we want to speak about Project Energy, the development of a targeted drug for the treatment of cervical cancer with a mechanism of action that affects the energy metabolism of a cancer cell. It's not a secret that cervical cancer takes 7th place in the structure of malignant neoplasms. It has been proven that HPV is the main factor in the tumor transformation of cervical epithelial cells. However, the mechanism of this transformation remains unexplored. Current trend in oncology is the revision of theories of oncogenesis. Nowadays, scientists around the world have returned to the study of energy metabolism of cancer cells. However, there is still no consensus on how the metabolism of a cancer cell changes. Recent published studies have shown that mitochondrial respiration and glycolysis in cancer cells sharply depressed, but the cancer cells continue to live, receive ATP, and what's the strangest thing, to divide aggressively. The question arises, which method of obtaining energy supports the vital activity, growth, and division of a tumor cell? The project team have developed the concept of energy metaplasia of cancer cells, the acquisition of an additional autotrophic method of energy production during oncogenesis, chemosynthesis.
Now we will briefly talk about proof of concept. The proposed concept was proved in 5 stages on a HeLa cell culture in more than 250 experiments. The studies were conducted in GMP laboratory. Analytical review of evolution of energy production methods by animals and plant cells revealed a possible chemosynthetic reaction in cancer cells, oxidation from iron ions from 2 to trivalent. At the second stage, we conducted the classical experiment of Vinogradsky, of iron binding in the culture medium of HeLa cells, thereby blocking the chemosynthetic reaction in 37 experiments. We received a sharp inhibition of the growth of malignant cells until their death. The experiments performed on the seahorse cell metabolism analyzer revealed in HeLa cells inhibition of 2 classical ways of generation energy, oxidative phosphorylation and glycolysis. We also conducted a bioinformatics study which revealed the presence in the human genome the homologs of bacteria genes responsible for chemosynthetic reactions. In vitro experiments using plant origin chemosynthesis inhibitors revealed a blocking effect on HeLa malignant cells culture. So, on the slides we see the data of an inhibitory effect of chemosynthesis blockers to malignant HeLa cells. In the groups without the addition of substances, as well as in the control groups of MSCs and fibroblasts, this effect was not observed.
The protocols of all experiments were published. Our findings open up a completely new approach to the understanding of oncogenesis, as well as the prevention and treatment of cervical cancer, the use of effective inhibitors of chemosynthesis. The purpose of Project Energy is to create a targeted drug for the treatment of cervical cancer with a mechanism of action that affects the energy metabolism of malignant cells, blocking chemosynthetic reactions. So, where we are now? The project team found a new mechanism involved in the growth of tumor mass in cervical cancer. The presence of chemosynthetic reactions in malignant HeLa cells was investigated and proved in vitro. Based on the results obtained, a strategy was chosen of detection of control protein specific only for this type of reaction and development of monoclonal antibodies to this protein. Such the control protein was discovered, and at the moment, together with Prime Biomed company, a monoclonal antibodies are being developed that binds this protein, which is responsible for electron transfer in the transport chain of chemosynthetic reactions. The first stage of the development has already done, namely cloning, expression, and purification of the recombinant control protein. After completing the second stage of development of monoclonal antibodies to the selected protein in the first quarter of 2020, it is planned to conduct advanced in vitro experiments on cell cultures and in vivo experiments on laboratory animals and prove the effectiveness of the prototype. IP protection is also planned.
After the completion of preclinical trials, it's planned to begin negotiations with the large pharmaceutical companies. Developed monoclonal antibodies can potentially be implemented as anti-cancer drugs for the treatment of cervical cancer. The global cervical cancer treatment market was estimated at $6.3 billion in 2017 and is expected to expand by an average of 5% over the forecast period. The global market for cervical cancer treatment, according to a new report, will reach $10.6 billion by 2025.
The project team consists of 5 founders, including 2 professors in oncology and gynecology sphere, a molecular biologist, and 2 biotech entrepreneurs with 5 years experience of commercialization of biomedical projects. Thank you for your attention.